Hello, everyone, and thank you for joining us today. I'd like to welcome all of you, those online and here in person, to today's presentation on focused ultrasound for temporary modulation of the brain. A quick note for those of you on the webinar, if you'd like to ask a question of Dr. Aubry, please submit it via the chat function at any time during the presentation. We will collect these questions until the Q&A period begins at the end of the presentation. We'll try to get to as many questions as we can during the time we have here. And now I'd like to introduce Dr. Jean-Francois Aubry, who is the Research Director at CNRS at the Institut Langevin in Paris, France, and he's a recently elected President of the International Society for Therapeutic Ultrasound. In 2012, Professor Aubry was awarded a Focused Ultrasound Foundation-funded fellowship, which allowed him to spend a year as an associate professor in the Department of Radiation Oncology at the University of Virginia, and he worked very closely with us at the Focused Ultrasound Foundation, so it's great to have him back here with us today. He's a leading ultrasound physicist who has made many significant contributions to the field of focused ultrasound, particularly in its applications in the brain. So Jean-Francois, take it away.
Thank you very much, Jessica, for the introduction, and thank you for giving me the opportunity to give this webinar. Before I start, as we might have a broad audience, I would like to give a very brief introduction of focused ultrasound. Well, similar to using a magnifying glass to concentrate light, it is possible to use focused ultrasound transducer to concentrate ultrasound energy at focus and perform a treatment. Here is an example for breast. Now, that being said, of course, the brain will be a very promising target for ultrasound and for MR-guided focused ultrasound. Nevertheless, there is a big challenge over there, and the challenge here on this picture is not what you see. You can see an image of the brain. The challenge is what you don't see, and what you don't see here in between the brain and the skin is the skull. That gives no signal here on MR, and actually the skull is distorting ultrasound waveforms. So how can we focus energy through the distorting skull bone in order to target the brain? As you can see over here, here is an example showing the effect that the skull is having on an ultrasound beam. And actually, so part of the energy is reflected, but it's also refracted by the bone and distorted. So at the end, after crossing the bone, the energy is spread over the brain, and it's not possible to focus the beam as tightly as I showed you before for breast, for example. How to do that? One idea is to compensate for the effect of the skull. So if no correction is applied, then the beam, you can see here, is distorted by the skull, and the peak pressure at focus is rather small. We could apply phase shift, and so send a distorted waveform. This waveform will be distorted by the skull in a way to restore the focus and get a high pressure at focus. The physics behind this is simply the wave equation and how waves propagate. So everything about this, and even applying shifts to compensate for that, is known since the 19th century with the D'Alembert wave equation. The thing is how to determine the appropriate time shift in a non-invasive way, and this was the challenge that had to be solved. One way to do it, and currently the only way, is to use to simulate actually the propagation of the waveform to the skull, and this can be done thanks to CT scans. And this D'Alembert equation here, in fact, you can from those CT scans feed this equation with density, speed of sound, and absorption coefficient to fully model the propagation through the skull. And here is an example of a simulation simulating the way that the the wavefront is distorted when going through the skull, and as we can predict how this wavefront is going to be distorted, we can deduce from that the phase shift that need to be applied to restore a good focusing through the skull bone. So you have an example on the left is without correction, on the right with correction. So it is possible to non-invasively restore the focus. So it took quite a long time, 2 centuries, to make this possible, and this has been made feasible thanks to 2 major technological breakthroughs, mostly in the 80s and in the 90s, sorry, and during in the last 10 years. First is the development of multi-element arrays, not one single element array, but multiple elements, because you need to be able to drive all those individual elements in order to apply the phase shifts and varying phase shifts along the skull to restore the focus. And the other is the increase in computational power. We need a lot of computational power to be able to simulate this wavefront over a large volume over the entire head. And not only this, but also increase in computational power in terms of imaging quality to allow a precise targeting. To illustrate this, let's have a look at how the imaging quality has been linked to computer power. By going back to the 80s, here is an example of the video games that were available at that time for tennis, and you see how it evolved in time, and now with almost virtual reality, you can see that the image quality rendering improved a lot with real-time 3D rendering. And the same, all this computer power has been also used in imaging to improve the quality, and the GPUs that are currently used in the video game industry are now used in the ultrasound scanners to improve the image quality. So now we have the possibility not only to model the focusing and the propagation of the wave through the skull thanks to the computers, but also we have access to very precise imaging of the brain with MR, and you see also that the ultrasound imaging quality improved a lot. So all combined together, here is an example of a brain prototype that we constructed in Paris in collaboration with the supersonic imaging. And you see here an image of the brain, and at the intersection here is the target that we choose to do the treatment. So based on the MR images, and you will notice that previously I showed you an image where you could not see the skull. The skull appeared as black, and you can see here details of the skull, because in fact this is a fusion between MR for the brain and CT images for the bone. So we have here combined access to MR for anatomy and CT for aberration correction, so that once the target is selected, here is the red point here at center, so based on the anatomical MR image, the software defines the best location of the transducer array in blue over there. And once this position has been selected, mostly perpendicular to the skull surface, then the software is computing the propagation of ultrasound between the target and the transducer, and the computation is achieved by using the CT data. Once the computation is done, we know what phase shift need to be applied, and here is an example of the refocusing that we can obtain. Here was an experiment on cadaver heads to test the accuracy of the system, and with dedicated MR images, we can measure the temperature elevation in the brain, and we compare the maximum temperature elevation on this image to the red point, to the actual target that was chosen. And as a matter of fact, the precision is linked to the size of the MR temperature voxel that was used. So the aberration correction now is accurate enough so that we can target the right voxel. and this has been published in the Journal of Neurosurgery. At the same time, Insightec was also developing a device, and it is currently used in clinics. I will show you an example later. And it consists of a multi-element array, again, with 1,000 elements that are operating at 700 kilohertz. And they are able to concentrate the beams through the skull, same way to target the brain. And here is a nice illustration and a treatment that has been performed at University of Virginia for essential tremor. 15 patients were enrolled, and they had medication-resistant and quite disabling essential tremor. And this was a feasibility and safety study. First, let's talk about the targeting. So here is the target that was chosen, and this is the area responsible for the tremor. It's exactly the same target I showed you previously in red, which shows the same target to verify the accuracy of the treatment in all systems, and this is with the Insightec system. So here is the targeting, and here is one patient before the procedure. So you can see that essential tremor is a disease that induces hand tremor, and it affects a lot the quality of life of patients, as patients cannot drink or eat by themselves, or they cannot even button a shirt. And same for handwriting. This is affecting a lot the handwriting. Here is an example where prior to the treatment, in order to test and quantify how the tremor was affecting the handwriting, patient was asked to draw spirals in here along those lines and straight lines here in between the gray area. So this is before treatment. This is, as I explained to you previously, the target for focused ultrasound. The patient goes into the MR. During all the procedure, the patient is awake. There is no incision, no electrode implemented. Currently, the treatment is consisting introducing an electrode for deep brain stimulation, so opening the skull and inserting the electrode. With focused ultrasound, there is no electrode, so no incision, so there should be no infection or brain damage because of the insertion of an electrode. So this is right before and right after the treatment. Here you can see a small dot over there, and this is the lesion that has been achieved with focused ultrasound. And right after the treatment, the patient is asked to draw the lines again, and you can see a dramatic improvement in the hand writing. And a few months after the procedure, so this patient was asked to transfer beans from one container to another, and you can see difference between before and after the procedure. And now the patient can eat and drink normally. So this procedure is quite a non-invasive life changer for those patients. And this study showed 75% reduction in the hand tremor over the 18 patients. And this is not the only study that has been conducted for treating essential tremor. There's been also a study in Toronto showing 80% reduction in the dominant hand tremor, and also in South Korea, in Seoul, showing again 70% tremor reduction. So on average, 75% hand tremor reduction for all those studies. So those are very promising results for focused ultrasound. Coming next is to expand those thermal treatments to other indications, to Parkinson's disease, to brain tumors, to epilepsy. And Parkinson's disease, a first case has been published by achieving a tracheotomy here on one patient, brain tumors as well. You can see here on this picture lesions, lesions that have been achieved within a tumor. And concerning epilepsy, this is on its way. Several targets have been defined depending on the type of epilepsy, and they are currently IDE under review at FDA to perform those treatments. So what could possibly be next, as there have been very interesting use of focused ultrasound for thermal ablation, and there's a lot coming next with thermal lesioning. But the one thing with ultrasound is that we can adjust the power, and there are many other ways to interact with the brain. The first one is the one I presented you before. It's called high-intensity focused ultrasound, where you can go all the way up to increase the temperature above a given threshold, and you literally cook tissues. And we were currently cooking brain tissues to treat essential tremor. So this is one way to interact with the brain, but we can also decrease the power, decrease the sonication duration, adapt the parameters to achieve other bioeffects. Of course, one very light effect is just by listening to the tiny echoes that come from tissues, and thanks to this, we can achieve ultrasound imaging. So this is one, the lighter effects interaction that we can have with tissues. Another interaction is the radiation force. So instead of sending very short pulses and listen to the echoes to perform ultrasound imaging, one can use an ultrasound transducer and sonicate for a longer time. 100 microseconds is enough to push tissues and to exert a force on the tissues and move tissues away from their original position. Here is an illustration at the surface of water. We can see that water is pushed up by the focused ultrasound transducer. And what you see here in water, you would have almost the same in tissues, except that the displacement would be much smaller, a few micron displacement. But those micrometric tissue displacement can be tracked with an ultrasound scanner, and you can reconstruct the elasticity of tissues, as this is equivalent to achieve remote palpation of tissues. It gives you access to the elasticity of tissue. It's a new modality that has been introduced a decade ago, and that is now implemented on some ultrasound scanners, and one commercial scanner from it is the supersonic imaging device. It's a good example of that. So could we achieve remote palpation in the brain as well? Well, we should. The thing is, we cannot use ultrasound to track this displacement because of the skull bone again, but we can use MR. And here's an example on a cadaver head of a displacement that has been induced in the brain and that is measured with dedicated MR sequences. So this micrometric tissue displacement can also be tracked with MR sequences, and it can be used, for example, for target verification in the brain to verify that the target is at the right location before switching to a very higher power and ablate tissues. It could be used, though, there might be other applications just to achieve remote palpation in the brain and try to induce effects on the brain and investigate what other effects could be done in the brain. So radiation force is one other example of the bioeffects. I already told you about HIFU, where you go all the way to cook tissues. With high power ultrasound but short sonication, it's also possible to destroy tissues, and namely this is used currently for kidney stone destruction. It is called lithotripsy, where you can break stones in the body. Instead of targeting stones, if you target tissues, then you can literally liquefy tissues, and this is called histotripsy. So basically, there are two main domains. One is interaction with tissues, but without any effect, long-term effect on tissues, imaging, and elasticity imaging or conventional imaging, so conventional imaging or remote palpation, let's say, and destructive effects that can range from thermal effects to mechanical effects, and those are permanent effects. And in between is what I'm going to talk about right now, and those are reversible effects. So you can change tissues, but not permanently, and in a reversible way. And I will mostly talk about what you can do on the brain, and that is reversible. The first example is blood-brain barrier opening. The blood-brain barrier is a barrier that protects the brain. Basically, it prevents viruses from going from the blood flow to the brain, which is good to protect the brain. The disadvantage is that it also prevents 90% of the drugs to go to the brain. So it means that to treat the brain, only less than 10% of current commercialized drugs are available for brain. And by using pulse ultrasound in combination with contrast agents are microbubbles that can oscillate with ultrasound, and they can stretch out the blood-brain barrier and open it and give access to drugs to the brain. And here is an example. This system is much simpler than the human brain system I presented you before. It's a single element transducer that can fit into an MR and can be used on rodents. And by using this device, you see here a difference between image of the brain before the opening of the BBB and after opening the BBB and injection of an MR contrast agent. So basically, if you have a closer look close to the eyes, you see a strong increase in the contrast close to the eyes, because the MR contrast agent could go into all those tissues and in muscles, but there is no contrast enhancement in the brain because it's protected and it prevents the contrast agents to go to the tissues, except at the location here where ultrasound was focused, and thanks to the combined action of ultrasound and microbubbles, the BBB was open and the contrast agent could go into the brain, and so would drugs. If you wish to learn more about this, I recommend you to watch the webinar that Elisa Konofago gave this year, and that is available on the website, so I won't expand on BBB opening, but it's a very promising technology, mostly for Alzheimer's and for brain tumors. Second effect is ultrasound neuromodulation, how to modulate brain activity with ultrasound. And why would we intend to use ultrasound for brain stimulation? There's already lots of ways to do so on the market. You can use surface electrodes or implanted electrodes for deep brain stimulation. It's also possible to use transcranial magnetic stimulation, or most recently, optogenetic to also stimulate the brain. But if you look at the characteristic of all those techniques, they cannot have both a good spatial resolution, be non-invasive, and be able to target deep-seated structures. But ultrasound has the potential to have a good spatial resolution, to be non-invasive, as I showed you before when focusing through the skull, and to target deep structures, as well as structures that are close to the surface of the brain. And here is the potential of focused ultrasound to get an idea of the size the spot that you can achieve with focused ultrasound. In general, when using a transducer that has a diameter D here, this is the aperture of the transducer, focusing at a given depth, F is the depth of the focus, then the lateral dimension is given by the diffraction theory, and it's the wavelength multiplied by the ratio of the depth over the aperture of the array. And in the elongation dimension here along the axis of the transducer is 7 times the wavelength multiplied by the same ratio but squared. So basically, when using a 300 kilohertz transducer, you would get a 5 millimeter large focus and around 2 centimeters in depth if F and D are equal, which is mostly the case for most transducers. And if you go all the way up to 1 megahertz, then you will get a millimetric precision. Ultrasound modulation of brain tissue, though, is not new, and it has been investigated and shown in the past. The Fry brothers have been the first ones to show evidence of ultrasound neuromodulation, and they showed, here is an experiment that they did it was published in 58 on cats, where they were in fact showing that they could induce reversible reduction of evoked potential as a response to light stimulation. You can see here in front of the cat the light stimulator. So they were showing by recording with electrodes the evoked potentials, and they could see reversible reduction. They could see also permanent reduction if they were increasing the power, but at lower power, they could induce reversible reduction of those spiking here, the evoked potentials. Ballantyne in the 50s also he sonicated ultrasound in one particular area of the brain of the cat, which is the Edinger-Westphal nucleus, and he could see either temporal dilation of the pupil for 1 hour, and then it would go back to normal, and permanent dilation if the applied power was too high. Lel in 63 also showed same kind of effects, but previously was on brain tissues, and he demonstrated it on peripheral nerves, and he was sonicating nerves. You can see here, this is the black line over there, where he would focus ultrasound at the nerve location, and he observed different types of effects. With high frequency ultrasound, again, they would see a near reversible change and inducing a depression of action potential along the nerve, whereas low intensity focused ultrasound were increasing, in fact, the conduction velocity and increasing the action potential amplitude. Moderate intensity was inducing a depression, but a reversible depression of action potential. So again, here, those studies show that we could switch from irreversible tissue damage to reversible tissue damage by decreasing the intensity. In the 80s, Adrianov, Tsironikov, and Gravilov showed also a lot of evidence of the interaction, reversible interaction of ultrasound with tissues, and here's an example for peripheral nerves, and this is in fact an experiment where they were focusing ultrasound here in the hand at various locations under the skin, and they were measuring thresholds for inducing a variety of sensations, tactile sensations, pain sensations, but also temperature sensations, and interestingly, they could induce warm or cold sensation. So not only warm sensation. So it's not ultrasound are not heating up tissues and feeling warm. It can also induce cold sensations. So those are also very strong evidence for reversible interaction with tissues. And for the rest of my talk, there's been also work on the retina to achieve neuromodulation on the retina. I will now focus mostly on the brain itself. And this interesting study from Tyler's group in 2010 made a big change in the field, and at least in our lab, we were very interested in that work and quite surprised as well. Tyler showed that contrary to what was done before, where they were decreasing the intensities and could see reversible change, Tyler used, I would say, extremely low intensity, extremely low energy, and they could see, they could trigger EMG responses with ultrasound, but with very low intensity ultrasound. You can see here that intensities were 0.2 watt per square centimeters, which is extremely small. And what was even more puzzling is that you see on this graph that the lower the intensity, the higher the effect. There seem to be no threshold, and by decreasing the intensity, the effect is just stronger, and you can hardly see an effect for high intensities and low frequency. This is the frequency axis. So in this case, low frequency, low intensity was the most efficient. And this rose a lot of questions in our lab, and we were wondering how we could get such an effect with such low intensity, and how this effect could increase when intensity was decreasing. And almost at the same time, Brigham and Women's Hospital, Yo and his colleagues showed also quite low intensity effects. Here's an example on a rabbit, and you can see 4-pole movement. And they tested quite a wide range of intensities, burst duration, pulse repetition. So they fine-tuned the acoustical parameters and showed that to elicit a 4-pole movement, they needed quite, well, there was a threshold, and they needed the average intensity to be higher than 6.3 watt per square centimeter. So in their case, in this study was showing a threshold for inducing a movement. So we were wondering, so is there a threshold, is there no threshold, is it that hard to induce neuromodulation? So we decided to conduct an experiment in Paris on rats. Here is the setup. We used a 300 kilohertz transducer that could be mounted on the rat head, and we anesthetized the rats. And of course, if anesthesia is too deep, it's possible to go all the way to very high power ultrasound and completely, in fact, cook the brain, and the rat would not move because it's deeply anesthetized. So in order to see a slight effect, you have to wait until the rat is almost waking up, so it stays still, but it's not deeply anesthetized. So the work window is quite small, only 15 minutes for one experiment, and then comes the wake-up phase. So we used pulse tonications at 300 kilohertz, and we were varying the pressure between 0.4 and 0.9 megapascal, and actually we saw a threshold corresponding to an average intensity of 7.5 watt per square centimeter. So in fact, pretty similar to what Senshikyo in Brigham and Women's Hospital was showing. But in fact, not only could we induce movement, illicit movements on the rats, but you will see that here is first example of movement of the tail that is induced on the rat when applying ultrasound, and it would stop right after the ultrasound pulse. We're going to increase a little bit the power, and you see a stronger movement. So I'm stopping here. So if you increase the intensity, the effect is stronger, and if you decrease the intensity, at some point there will be no effect anymore. So this is how we could set up the threshold to see actually a movement. But depending on where the ultrasound transducer is located, it's also possible to get other effects. And here is an example here where you can see an eye movement, and with this location, the transducer, the tail is not moving anymore, nothing moves but the eye, and always towards the nose. And here is an example where only two whiskers are moving when we apply ultrasound, and there is no movement of the tail, no movement of the eye, only the whiskers would move in that case. And this was quite surprising, especially given the size of the focus. As I mentioned before, we know, given the size of the transducer, given the frequency that we're using, we can roughly estimate the size of the focus. And in our case, it was supposed to be 5 millimeters in diameter and 2 centimeters along the axis, which means that this almost 1/3 of the rat's brain. It's pretty big, but yet we could get very specific effects. And this was puzzling to us. So we conducted the simulations into the rat brain with the same program that we used before to simulate the propagation to the skull for humans, for brain treatments. And we did not use the software this time to determine the phase shifts, but to have a look to what the pressure is looking like in the brain. And here is the results of the simulation. So it confirms that the pressure is spread over a large volume of the head, but you can see that there are standing waves occurring and mostly reflecting on the bottom of the head of the animal. And so it gives quite a complex pattern, and we believe that this pattern is highly changing when we move the transducer. It's very difficult yet to know how we can target very specific area of the brain, but at least it makes sense that specific effect can be obtained in the brain, even though the wavelength is pretty big, and the most part of the brain is currently insonified by the ultrasound. So you can find more details in this article detailing both the effects that were obtained, the threshold, and also the simulations. So then next question was, how can we get a tighter focus to better control the field in the brain and not rely on any reverberation in the brain? One possibility is to decrease the wavelength, and so to increase the frequency. And so we were having a 5 millimeter spot. We can go down to a 1.5 millimeter with a 1 megahertz array. We could go higher up, and we would have then almost 40 microns in theory spots. But the problem is that when increasing the frequency, it has been shown by a study from a Stanford group, Kim Pauli and Ronnie King, that when increasing the frequency, the threshold for achieving neuromodulation is increasing as well. So there's still ongoing work to double check if this is such a strong dependence. As you can see here, by changing very slightly the frequency, the threshold is increasing a lot. But at least this is the trend. I mean, what is sure is that the higher in frequency you go, the most difficult it will be to get to achieve neuromodulation, and it's important to do it in a reversible way and not to damage tissue. So there are still work being done on increasing the frequency, but it's not as simple as it might seem. So in fact, there are two procedures. One, of course, we could increase frequency. What about increasing the brain? You can't really increase the brain, but you can switch from one species to another one and switch from rat to primate. And in that case, the brain case would be larger and more suited to the use of low frequency ultrasound. And this is what we did in Paris, where we used actually the same setup that was used for rats, the same transducer. You can see here mounted on an arm. And the only difference is that this was guided by using a neuro navigator and to be able to target specific areas of the brain. In our case, we were actually targeting the frontal eye field, and we showed that focused ultrasound was changing the reaction time of monkeys that were asked to perform a voluntary eye movement. So it was the first time that neuromodulation was achieved on non-human primates, and a few months after, Tyler's group also published a work not on non-human primates, on primates as well, but this time on humans, and they showed evidence that they could modulate brain activity. So I need to spend a few time explaining exactly what they did. So actually, there was a nerve stimulator attached to the wrist of volunteers, and this stimulator was adjusted to elicit very slight twitch of the thumb. And they were recording with ECG on top of the head the response of the brain to this excitation. So this is not achieved with ultrasound, it's achieved with a nerve stimulator, so electricity that is just adjusted to elicit a slight movement, so it's not painful, but it would elicit a movement, and they record the activity in the brain. And then, at the same time, there is an ultrasound transducer that is aiming at different locations of the brain, and what they showed is that the ECG recording was changing depending on the location where ultrasound was applied, with low power again, by only slightly modulating the activity. And this is a reversible effect, and the intensities used here for this experiment were much lower than the ones that are expected to induce permanent change. So it was the first time on humans that a team was able to show modulation of brain activity. And almost at the same time, a set of experiments were conducted in South Korea. They were published a little bit later, but they were achieved almost at the same time, where they did not modulate brain activity, but they stimulated brain activity. So in that case, ultrasound are targeting the almost the same area of the brain than before, the primary somatosensory cortex, and no electric pulse in the hands. I mean, the hands have nothing, but they could elicit sensations in the hands by focusing ultrasound on the brain surface. And they elicited sensations in the hands of 11 out of 12 volunteers. And it appeared that the only non-responding volunteer, in fact, had a thicker skull. So it might be that the the intensity in the brain when below the threshold for inducing a stimulation. So those two studies are very promising in switching from animal work to humans. Sorry, I don't know why it's not. Okay, so what are the mechanisms for ultrasonic neuromodulation? There is a very interesting work published by William Tyler and his group showing here on brain slices that calcium and sodium channels are open with ultrasound. And here is a movie. So when I'm going to play it twice, when the red point is appearing on top is when ultrasound is shooting, and when ultrasound are on, you see suddenly boom, a firing here of all those channels, and they are here imaging into those slices the calcium released. So according to them, ultrasound stimulates neuronal activity, and it is triggering voltage-gated sodium and calcium gates. And so they believe that this opening it's a mechanical effect, and it's opening mechanically sensitive voltage-gated channel. So that would be a mechanical effect on the brain. When Kim Pauli's group investigated the influence of frequency, in fact, one of the conclusions of that work, one is that it's harder for higher frequencies, and the consequence of that is that it might be merely fatal for the explanation of a radiation force imaging, so direct action potential triggering by just achieving remote palpation in the brain, and they believe that this is rather consistent with the appearance of cavitation into the brain. This was quite surprising because the intensities used are lower than what is currently known to induce cavitation. Nevertheless, there's been an interesting modeling of the possible interaction of ultrasound with brain tissues by Shai Shoham and Eitan Kimmel in Israel at Technion, and here is they assume that on the B layers, bilipidic layers that are from which are made the neurons, that the oscillations and expansions and contractions of the intramembrane space is in fact having the membrane to act as a capacitor, and as it expands and contracts, it accumulates charges, and it can trigger action potential if ultrasound sonication is achieved long enough. And basically, they assume that there is some nanocavitation, picocavitation occurring, and that this bubble is oscillating and growing very slowly, and having the membrane to oscillate until it triggers action potential. And this is most probably what is happening in the brain. So, as I showed you, it's possible to switch from no effect on tissues to destructive effects, and in between is an area where we can have reversible effects in the brain that are not dangerous, that tissues go back to normal. Of course, the FDA administration gave threshold for those. So, namely for destructive effects and destructive thermal effects can be quantified with basically with the intensity that is being used. Whereas for mechanical effects, for destroying tissues mechanically, there is an index that is called the mechanical index, and this index should not be higher than 1.9 in order to be able to use ultrasound as an imaging modality. So this area here is completely safe. Here is reversible effects that are currently more and more investigated, and over there are the destructive effects. Quite interestingly, the Food and Drug Administration, of course, is very preservative, and those numbers are actually quite low. And the publication on the bottom is proposing that in some cases it could be possible to increase the intensity of the mechanical index without putting the tissues into the danger zone. There is some margin into it, and I quite agree with that. And so this capacity to fine-tune the ultrasound parameters from no effect to reversible to destructive effect, in fact, is the basis of our project in Paris. It is called Brain Therapy, Brain Neuromodulation, and Functional Imaging with Ultrasound. It's called the UltraBrain Project, and it's been funded by the French Agency for National Research and the Bettencourt-Schuller Foundation. It's a collaboration between our lab and the Institute for Brain and Spinal Cord, Institut du Cerveau de la Moelle, ICM in Paris. So I showed you that by using continuous sonication with quite a high intensity focused ultrasound, HIFU is possible to induce thermal lesioning and achieve thermal therapy. By using pulsed ultrasound in conjunction with microbubble injection, it is possible to transiently and very locally open the BBB with using pulses of ultrasound and no contrast agent ultrasound alone. It's possible to induce ultrasonic neurostimulation and elicit movement, for example, on animals, or trigger action potentials. And lastly, also, we intend to use ultra-fast functional imaging to image the brain, and Michael Tanter in Paris is in charge of this, and this is becoming a very huge application in our lab to image the brain with ultrasound. And for each of those indications, I presented you before the rodent system, so we can achieve rodent experiments, basically to test the efficacy and the safety of the approach. And we also have access to primates for either with the small system for neuromodulation. I showed you the handheld system, but we also have the preclinical system that we can use on primates to test for thermotherapy, and the endpoint is for humans, to go for humans for each individual application. For humans, for thermotherapy, we will go for essential tremor and metastasis treatments. For BBB opening, glioblastoma treatments. For ultrasonic neurostimulation, it will help, hopefully, to guide essential tremor treatments, as well as we hope to treat depression with ultrasonic neurostimulation only and no thermal lesioning. And finally, on humans, it has already been done, as a matter of fact, here for brain imaging with ultrafast imaging during operation, so it's per-operation imaging. I would like to thank the medical team at Institut Langevin who mostly contributed to this work, and also, of course, from ICM, Pierre Pouget and Nicolas Vattiez, with whom we are currently collaboration on the primate experiments, and there's still a lot of research going on with them currently. I would like to give some credit for some of the pictures that I presented you, the FEST Foundation, and also Jeff Elias and Rob Dalla Piazza, neurosurgeons at University of Virginia, for the essential tremor results that I showed you, and Natalia Vikotseva for some of the pictures I showed you in the history of neuromodulation. Of course, we thank the ANR for supported our work, as well as the Bettencourt-Schuller Foundation that has been extremely supportive for our work. If you want to know more about therapeutic ultrasound, I invite you to save the date for 2017. That will be the Winter School on Therapeutic Ultrasound in France, in the Alps. And next is the 2016 ISTU Symposium that will be held in Israel, and we will be very happy to welcome you and discuss not only about neuromodulation, and not only about the brain, but about all the applications of therapeutic ultrasound worldwide. Thank you very much.
Thank you. That was great. First, I want to address, or see if you can address, a comment that we received from someone on the webinar, indicating that, or suggesting that using a range of frequencies centered around 320 kilohertz could help to eliminate aberrations and standing wave effects. Using a chirp or random signal ranging from 300 to 340 kilohertz, for example, may be possible.
Yes, so it is true that it would, it could eliminate standing waves, but the spot would be rather large anyway, because the standing waves, I don't think they are enlarging the spot. It's just that, in fact, the spot is even tinier with standing waves, because we see stripes on the image, whereas by using chirp of a random signal, it would be more homogeneous, but it might be also larger. So it would be very interesting, though, to test how this would change neuromodulation. For example, it might also, so it will eliminate standing waves. I just don't know if standing waves are useful or not in that case. So maybe it would also eliminate the effect. I have no idea, or at least those very specific effects. So maybe in that case, you would have both the tail and the whiskers and the eye moving at the same time. I don't know what could happen, but Christian is right. It would eliminate standing waves. I just don't know exactly what would be the consequence in the brain.
A quick question: what do you see as the biggest challenge as neuromodulation is moving towards clinical use?
I think it will be adjusting the power, making sure that the power, the acoustic intensity in the brain is the right one. Currently, in the study that has been conducted in Seoul, in South Korea, they show that one of the volunteers was not responding, and by having a look to the data after the experiment, they noticed that the skull was thicker, so that the intensity was less than they expected, and it might be below threshold. So it means that it seems easier to penetrate the skull with those low frequency devices, but to be able to predict the intensity in the brain and adjust to the right intensity would be the challenge, because you don't want the intensity to be too high in the brain, so that you will not damage tissues for someone who has, for example, a very thin skull, and you want to see an effect, even for patients who have a thick skull. So it needs to be an adaptive focusing technique yet, just to fine-tune the parameters. And also, currently, there has been work showing evidence that one set of parameters is working, but it has not been optimized yet. So we don't know what is the optimum set of parameters for a given frequency, what is the sonication length, the pulse duration, the repetition rate that should be applied. This is currently, maybe it might be a little bit too early to go to humans, and this fine-tuning of the parameters is what we are currently doing on primates first, to be able to span over a large range of intensities, pulse repetition, frequency as well. This takes a lot of time and a lot of experiments to test each individual parameter.
Another question from our webinar audience. Other than intramembrane cavitation changing the capacitance values, could sonoporation and the accompanying reversible perforation of the membrane increase ion flux and likelihood of depolarization?
That's a good question. Well, yeah, it could. Most of the time, though, sonoporation is a lot more efficient when used in combination with contrast agents. And for neuromodulation, there is no contrast agent injected into tissues, so sonoporation might be less active. But yet, it could be the case. I think also radiation pressure could also wash out neurotransmitters in between synapses. So I could also understand that it could be a way to, I would say, to decrease brain activity. That's also, I think there's been some evidence a long time ago, and this should have to be double checked currently. But the exact mechanisms, though, are not known so well. I think the best paper, the most intensive in terms of experiments, is the one from Tyler's group on slices of brain, and the intramembrane cavitation. It's very appealing, but it's a theory, and now we would need to have experiments demonstrating that this is actually what is going on. And I would say, especially for investigating neuromodulation mechanisms, we need to work now in collaboration with neurobiologists and biologists to be able to see what is happening at the cellular level. And it's not that simple, because even the wavelengths we're using, we are not actually having an effect on one single neuron, but this is a very large scale effect that is happening in the brain.
Another question here. Does the modulation threshold change for different areas of the brain?
We were not able to see a change when we were changing locations in the brain, and I don't think we are precise enough to answer that question. It could be the case, though. We know that for thermal ablation, the thermal rise strongly depends on where you sonicate in the brain, and white matter and gray matter have different absorption coefficients, so that different areas of the brain respond differently to ultrasound in terms of heat dissipation. And so that could also be the case for neuromodulation. Again, this has to be investigated, and I don't think anyone already showed. People changed locations to see different effects in the brain, but I've not seen any reports of a different threshold. And it's also difficult to estimate what is the exact pressure in the brain. If we think of rat experiments, I showed you the reverberations in the head, and by estimating the maximum pressure in the brain is not easy, and we're not 100% sure that the part of the brain that are currently triggering action potential correspond to the maximum pressure in the brain. So we maybe we need some microscopy experiments to be able to differentiate from one neuron to another one. I know within the theory from the Eitan Kimmel group, they also expect that excitatory neurons would have a different threshold than I would say inhibitors than inhibitors, yes, than inhibitory neurons. So in this case, at least we would have two kinds of neurons that should not have the same threshold. They would like us to test it. It would be nice to test if this is the case, and they do not respond the same way to ultrasound, and obviously inhibitory and excitatory neurons do not have the same dependence on the duty cycle that is currently used for ultrasound.
So do you think that the technique can be precise enough for brain mapping?
Yes, I think so. I think this is one of the next steps, and to combine ultrasound neuromodulation to activate the brain, to brain imaging, and see actually what part of the brain is responding to a solicitation that is being applied to another part of the brain. So connectivity mapping.
Another comment from our online audience. In your study regarding pressure field distribution, 40% of your cases were not successful. You evaluated the anesthesia level by pedal reflux. It could be better to use longer-lasting anesthetics to keep the level of anesthesia more constant. Additionally, evaluate the level of anesthesia quantitatively using breath and heart rate. This could be important because all findings indicate that focused ultrasound neuromodulation is very anesthesia dependent.
Yeah, I fully agree with that. And we also noticed that in our case that first neuromodulation is anesthesia dependent, and actually in the paper we give two different thresholds, one for late stage anesthesia, so whether deeper or lighter anesthesia gave different thresholds for inducing effect in the brain. And in fact, one of the reasons also why we switched from rats to primates is that we could have awake primates achieving the task. So we wanted to get rid of anesthesia because it's very complex. When tuning the ultrasound parameters, playing with frequency, intensity, what we say pressure amplitude, pulse duration, repetition rate, that's a lot of parameters, and on top of that is anesthesia, so light or deep, or also depending on the kind of anesthesia that can be used, and one last thing though, just about the comment about the 40% time the experiments were not working. We were not the only one to see that happening in our experiments. It's true that we were the only one to publish this and to actually say that we had only 60% of the experiments that were successful. But other groups, they just omit to state that, but it was the case. So it's not always 100% successful, at least when using ketamine, xylazine anesthesia, that's for sure.
Okay, I think we have addressed all the questions. So this concludes today's webinar. Thank you, Jean-Francois, again, and thank you all for joining us today. And stay tuned to our newsletter and website for invitations to future webinars. Thank you.